recording now. Now, this is the latest exam. Let me zoom in. March 2024, 9700 AS Bio. Okay, now here they have a diagram. It's a scanned image, unfortunately. This is what I could put my hands on. Now, this is a diagram. I'll try to zoom in so we can see better, representing part of the phospholipid bilayer of a cell surface membrane. And I identify the part of a phospholipid molecule labeled A here in figure 1.1 that forms bonds with the phosphate heads and with the fatty acid tails. So what is this part? A, what is A? This is what? They did give you, can you hear me? Yes, this is the ester bond. They did give you here some labels. They told you this is the phosphate head, and these are the fatty acid tails. I know it's not very clear. And this is the bond between the phosphate head and the fatty acid tail. So this is definitely the ester bond. Now let's remind ourselves quickly of the ester bond. In the case of these ester bond, you have a glycerol molecule, which is an acid. This is the hydroxyl group and the fatty acid tail. What are the two functional groups in fatty acids? They are a chain of hydrocarbons, and we have two functional groups. What are they?
COOH.
Yes, very good. The hydroxyl group. And on the other side, what do we have?
OH.
No. CH3, the methyl group. Okay. And then what happens? How do we create the ester bond? The hydroxyl group here joins together to give me what is removed? Water. This is a condensation reaction, and the bond is between the O here, and it is bonded to the carbon from one side and another carbon from the other side. So the ester bond looks like this. Fine. And one of the carbons is double bonded to an oxygen. This is the ester bond, whether in the triglyceride or the phospholipid. Okay, let's revise together, even if they did not ask. Now here, this is a cholesterol molecule, and they gave you the molecular structure. They told you there is a non-polar part and there is a polar part. We know that the cholesterol is just like the phospholipid. It has a hydrophobic part and a hydrophilic part. Using the information in the figure, explain the orientation or positioning of the cholesterol molecules in the phospholipid bilayer. How is the cholesterol molecule also positioned in the phospholipid cell membrane? What will you say here, Farouk?
The
Hmm.
polar group faces the water.
Yes, you are correct. The polar group, it will face the watery environments. Okay. And what about the non-polar?
It will face the other side, away from the water.
What do you mean the other side? What is the other side? We have a phospholipid bilayer. We have the phosphate heads and two fatty acid tails, and they are bilayer. So we have two of those that are facing each other. Now, the polar part of the cholesterol will be at A or B?
A.
Yes, so the polar part will be facing towards the water, or it will be close to the phosphate heads. Either both will be right, but facing towards the water is very good. And the non-polar part of the cholesterol will face towards A or B?
B. The fatty
Yes.
acid.
Very good. So the non-polar part of the cholesterol will be towards the fatty acid tail. Do not say, like you just told me here, that the non-polar part will be towards the other side. This is not something we write on an exam. What is the other side? You mentioned the side clearly when you talked about the polar end. The polar end of the cholesterol will face the water, or it will be close to the phosphate heads in the phospholipid bilayer, which is the same as facing water. Okay, it will form hydrogen bonds with water, right? Everything you know. And the non-polar part, don't say it will face the opposite side. What is the opposite side? This side, away from the water. within the hydrophobic tails. So the nonpolar part will face away from the water, opposite of what you said, away from water, okay, or towards the hydrophobic center or hydrophobic core of the fatty acid tails, hydrophobic core of the phospholipid tails. Both of them they mean the same thing, but don't say it's going to face the other side. This is so not clear. You will not get a grade for that. You understand, Farouk?
Yes, miss. Thank you.
Okay. State, Mariam, one role of cholesterol in phospholipid layers. We studied many functions. Give me all the functions that you studied.
They can regulate the fluidity of the phospholipid bilayer.
Very good. What else? What other function do they have? The main function is the fluidity. I agree with you. But what if they ask for another function? They provide support, by the way, which is close to fluidity, like they mean the same thing. Because without the cholesterol, the cell membranes would break. What else? What else? They provide support, fluidity, huh? Because they have a hydrophilic part and a hydrophobic part, they also contribute in making the cell membrane what?
Semi-permeable.
Yes, semi-permeable. So they also contribute in helping the cell membrane become semi-permeable because they only allow polar or non-polar molecules to pass.
Non-polar.
No. If it's non-polar, it can directly pass across the cell membrane without any help.
Okay.
Yes,
Polar
it makes it
molecule.
semi-permeable. Yes, you are right, by the way. It makes it semi-permeable. It's not a channel protein. So it makes it semi-permeable because it also has a hydrophobic end and a hydrophilic end. So it only allows non-polar molecules. You are correct. Okay. Explain why sodium ions cannot pass across the phospholipid bilayer by simple diffusion. Very easy question. Why polar molecules or sodium ions cannot cross the phospholipid bilayer? Let me see, who do I have here? Kareem, Adham. Hello, Adham. Hello, Kareem. Can one of you tell me why sodium ions cannot cross the phospholipid bilayer by simple diffusion?
The molecules are too big.
It's
No,
charged.
Habibi.
Can't pass.
No, they're not too big.
No,
Yes, they are charged.
They are charged and can't pass the hydrophobic core of the phospholipid bilayer.
Of course, they are charged or they are polar. They cannot cross. Let me change color. They cannot cross. I'll write it down because I'll send it to you solved, okay? They cannot cross the hydrophobic core. Then what do they need, Adham, to cross the this is enough, but what if the question is out of 2 marks? What do they need so that they can cross the hydrophobic core?
Transporter proteins.
Very good. They need channel proteins or carrier proteins, and they're all correct, or carrier proteins. They just need proteins, transport proteins with a hydrophilic core so that they can cross the cell membrane. Okay, the next question. Now we have to read it. Prostaglandins are small lipids and produced in many tissues of the body. One role of the prostaglandins is to cause inflammation at the site of injury. Inflammation is the normal first response in the immune system to injury or infection. Cyclooxygenase is an enzyme. Mariam, you want to say something?
There is a question above this one.
Really?
Yes.
Oh, because we zoomed in. Thank you for telling us. 1 minute. Oh, yes, there's a big question. Okay. Compare facilitated diffusion and active transport by stating one way in which they are similar and two ways in which facilitated diffusion is different. Okay, this is also an easy question. What is the similarity between facilitated diffusion and active transport?
Both allow polar molecules to pass through the cell membrane.
Very good. So both of them, they allow the passage of polar molecules. Now, give me two differences. Kareem, can you give me a difference between facilitated diffusion and active transport? Very easy differences. This is an O-level question, Kareem. Adham. Mariam, can you continue?
The active transport, the molecules move from low concentration to high concentration.
Yes.
And in active transport, the polar molecules move through the carrier proteins and the transport proteins.
Give me another difference that is very obvious. Because facilitated diffusion can happen can happen across both types of proteins, the carrier and the channel. Active transport only carrier proteins. So if you want to mention that, you have to be precise. Facilitated diffusion, this is diffusion with help. So molecules will move down a concentration gradient using carrier proteins or channel proteins. Any type of protein will do. But active transport, which type of protein that is needed? The carrier or the channel? The carrier only. The channel protein, because they do not change shape, they will not fit here. Another very obvious difference.
Active transport
Which one?
conserves energy.
Of course, active transports need energy. Facilitated diffusion does not need energy. A third difference, very easy. Active transport, what about the concentration gradients? Very easy difference. We said it from low to high. Yeah, we said that. So we said the differences. Active transport needs energy. Facilitated diffusion, no energy. Active transport from low to high. Of course, facilitated diffusion from high to low. Facilitated diffusion needs a protein, whether carrier or channel. And active transport only the carrier protein that can change shape. Okay, this question. Let's continue. This enzyme will catalyze one of the steps in the reaction pathway for the formation of prostaglandins from phospholipids. The reaction pathway occurs in the smooth endoplasmic reticulum of cells. Part of the reaction pathway is shown. So this is the reaction pathway. I have a phospholipid, and then it will change into an acid, and then with the help of the enzyme, this acid will change into a prostaglandin. Suggest an advantage for this reaction pathway occurring in the smooth endoplasmic reticulum of a cell rather than in the cytoplasm. Again, a very easy question. Who will give me an answer? Tell me, who do I have here? Farouk. This reaction, why is it better to happen in the smooth endoplasmic reticulum, not the cytoplasm?
I don't know.
Come on, this is so easy. What is the function of the smooth endoplasmic reticulum? Pay attention to the question. We started with the phospholipid, which is the lipids.
Yeah. Oh, since lipids. Okay.
Then what? What will you say?
SGR is the place where lipids are synthesized.
Yes, the smooth endoplasmic reticulum will produce the lipid, will produce the phospholipid. So it's there. I don't have to, in the cytoplasm, no phospholipids are produced. Very easy question. Okay. Sometimes modifying the R group of an amino acid in the enzymes can reduce the catalytic activity of the enzyme. Suggest how changing the R group of an amino acid in an enzyme will reduce the catalytic activity of the enzyme. Now, this again is very easy. They're talking about the enzyme, and they said that aspirin will reduce the catalytic activity of the enzyme. What is catalytic activity? Even if you did not hear about it before, it's very obvious from the name. It's the activity of the enzyme. How will the catalytic activity of the enzyme, or just the activity of the enzyme, will be affected when the R group is modified? Now, this is a very easy question also.
Decrease.
You will write decrease. That's the only word you will write.
No, it will decrease because the The
And by the
amino
way,
acid
I
is
want to
no
stop you.
longer.
I'm sorry, I'll stop you. They already said in the question it will be reduced. So you did not give any added information.
Okay.
Mm.
Because the amino acid is no longer complementary to the active site of the enzyme to can form enzyme substrate
Exactly.
complex.
Very good. Okay. Now, something else. I want you to add more information. When you change the R group of the amino acid, what are you changing?
The shape,
Excuse me? What did you say?
the shape,
The shape of what?
the amino acid.
Yes, you are right. I want you to be sure of the answer. What you said is right, but I want you to add on to it. You said that when we change the R group, we will change the active site. Correct. It will no longer be complementary to the substrate. Correct. Okay. But you can get a 2 out of a 3 because you can write some other information. We know that the structure of any amino acid is a carbon in the center. Yes. Bonded to what are the two functional groups? Someone
For the
give me an
amino acid,
answer. Yes, for the amino acid, yes.
COOH,
Yes, carboxyl group at one end. And what else?
amine group on the other side.
And an amine group. Okay. And that central carbon is bonded to a hydrogen and a variable R group that determines the type of amino acid. Which type of amino acid? It is hydrophobic, it is hydrophilic, it is valine, it is methane, it is valine, it is methionine, it is whatever. So the R group determines the type of the amino acid. When we change the R group, we are going to change the type of the amino acid. When we change the type of the amino acid, then we will change the primary structure of the amino acid, the sequence of amino acids. We change the primary structure, we will change the secondary structure. We change the secondary structure, we will change the tertiary structure. So in the end, we will change the final protein with a different active site that is not going to be complementary with the substrate and not form an enzyme substrate complex and reduce the catalytic activity of the enzyme. Okay, so I just want you to write things in order. You told me, Kareem, Farouk, that the amino acid will have a different active site and it will no longer be complementary to the substrate. And you are right. Why will you take 2 out of 3? Because there is more information that you can add on and you didn't. You did not mention that it will not be able to form an enzyme substrate complex. You did not mention that changing the R
I
group.
mentioned the enzyme substrate complex.
You did? Good. Okay. We can add that changing the R group is going to change the whole amino acids. The type of the amino acid will change. Different amino acids, different sequence, different primary structure, secondary structure, tertiary structure, and everything is going to change from there. Okay. So if we want to write a model answer, we don't have the mark scheme. I couldn't find it, but always when you have questions like these, especially if they are 3 marks and above, write everything that crosses your mind so that you are sure that you get the full mark. You don't know exactly what they want. So let's write it from the very beginning in order that when the R group changes, it's going to change the whole what? It's going to change the type of the, wake up with
Amino
me.
acid.
Very good. So the amino acid will change. So the sequence of amino acid changes. This is going to change the primary structure, secondary structure, tertiary structure. And this is going to change the shape of the active site. One minute. What happened to my phone? It will change the shape of the active site. And no, it will no longer be complementary. And no enzyme substrate complex. It will not be able to carry out its normal reaction. It will not be able to reduce the activation energy. Anything that crosses your minds that relates to the function of the enzyme, write it down. Okay? Prostaglandins are example of cell signaling molecules. Outline the process of cell signaling that leads to a response by the cells involved in inflammation. Adham, what are the steps of cell signaling? Someone give me an answer and speak up.
Cells release vesicles.
No, they don't release vesicles. In cell signaling, first, the cells release something. What does it release? It's not a vesicle. What is inside this vesicle? Okay, what does it release?
Transmitters.
What is? Hmm?
neurotransmitters أو حاجة زي كده.
Yes, give examples. The cells will release a ligand. It's called a ligand. This ligand can be a hormone, a neurotransmitter, whatever. Okay, and whatever is released will then, what's the next step?
Will then be released outside of the cell to attach to the receptors.
Very good. And then we have reception. Or you don't have to write this. It was going to bind with a complementary receptor. This complementary receptor, where is it found?
On the cell surface membrane.
Very good. Of which cell or organ?
Which target cell.
Very good. Yes, a target cell or organ is found on a target cell or a target organ. And then what will it do when it binds to the complementary receptor on a target cell or a target organ?
Initiate a reaction.
Very good. It will initiate a series of reactions or activate a secondary messenger. So it will initiate reactions until we reach a final
product.
Yes, a final response. Okay, this is a very easy question also. Cell signaling, something is released, it will be received by a complementary receptor on the target organ, it will cause a series of reactions or activate enzymes, and then the final result or response. Okay, let's continue. Until now, I see that the exam is easy. Let's see, description. A large cell that has a bean-shaped, kidney-shaped nucleus. It can develop into a macrophage. What is the name of this white blood cell?
Phagocytes.
Which phagocyte? Because we have two phagocytes.
Yeah.
The neutrophil or the monocyte?
The neutrophil?
No, the neutrophil is large and irregular and it has a lobe nucleus. But which one is bigger and has a kidney-shaped nucleus?
The monocyte.
The monocyte. Don't forget, we want the monocyte. Okay? A cell that has a large spherical nucleus and little cytoplasm and it responds to non-self antigens. Lesson immunity. Which one has a large nucleus that occupies most of the cell? It looks like this. The lymphocyte or the monocyte or the neutrophil or the what? Come on. Immunity. Lymphocyte. Yes, whether the B or the T lymphocyte, just lymphocyte. Now, a cell that has a lobe nucleus and it is phagocytic. This one.
The neutrophil.
Yes. So in the first one here, you have to mention monocyte. They wanted monocytes. Don't just say it's a phagocyte. Which phagocyte? Okay. Now, we have here pictures of two things. We have to read the question. Now, camels are classified in the family of Camelidae and live in the desert habitats of North Africa and Asia. In these hot, dry environments, these camels can lose up to 30% of their body mass from dehydration, causing their blood to become more viscous or thicker. Figure 2.1 shows a drawing of red blood cells of a camel, and figure 2.2 is a drawing of a human red blood cell. So this is the red blood cell of a camel, and this picture is the red blood cell of a human. Of course, they will ask you, what is the difference? Let's read the question. Figure 1.1 and 2.2 show differences between the red blood cells of the camel and the humans. Suggest how these differences adapt camels for living in a hot, dry environment. 3 marks. Now, suggest questions are not nice. Suggest question, the moment you see a suggest question, understand it as out of syllabus. They're asking for a critical thinking question. They want your opinion, but your opinion has to be the same as theirs. So, these are the red blood cells of the camels, and camels, they live in the desert. Can you look at the picture and suggest how these red blood cells can adapt to dehydration, but the human red blood cells cannot adapt to dehydration? What do you see?
We can't see.
You cannot see? What do you mean you cannot see?
Like the thing is, I can't see the picture.
What is the
on the
screen not
screen.
shared?
It is, but it's too down.
Is this an answer that you are going to write on the exam? Who is speaking? Farouk or Adham?
Doctor, the
Okay,
picture was
I can see it now.
What did you say, Mariam?
the picture was lagging. It was between the 2 pages.
Ah, okay, fine. Now I understand. Now you can see the picture?
Yes.
Yes.
Okay. You have to think of something, whether you are convinced or you're not convinced. What do you see in the red blood cells of a camel that makes it more adapted to the red blood cell of a human?
It's less turgid.
Less turgid? How do you know it is less turgid?
It looks like it's not as full. I don't know.
Habibi, I cannot hear you. Can you put up your voice?
It's like it doesn't look as full of water. I don't know. I'm guessing.
I still cannot hear you. Can someone hear him? I cannot hear you. I did not understand what you said.
It's fine.
Said it looks less turgid.
Okay, how does it look less turgid? How did you figure out that it looks less turgid? Let's make a table, and we will compare the red blood cell of the camel with the red blood cell of the human. Now, what I can see, the very, very obvious difference, that the camel red blood cell is oval, so the difference is very obvious in shape. And that of the human, what is the shape?
Biconcave.
Biconcave. This is an obvious difference. So the camel, it has an oval red blood cell, and the human red blood cell is biconcave. And you think of how the oval red blood cell is going to help in dehydration? So one, it's oval.
Squeeze
Think
more
of something.
efficient
More efficient in doing what?
in squeezing in the blood vessels.
How
Or
will
maybe
this
larger
help
surface
in...
area, maybe larger surface area, so it can
Yes, you're close.
get more oxygen.
Forget the oxygen, Habibi. We're talking about dehydration and storing water.
Storing water, more surface area,
Yes. Yes, you're
more
right.
aquaporins, so more water attraction.
What did you say? What was the last thing you say?
More aquaporins, so attracts more water.
No, leave the aquaporins now, because the aquaporins does not attract water. Aquaporins, these are channels that allow the movement of water. So leave it now. So we obviously see the difference in shape. The camel has an oval red blood cell, but the red blood cell of the human is biconcave. Adham said that this will increase the surface area. Yes, it's obviously a little bit bigger, or the shape will give it more surface area. And the oval red blood cell, you can say it's larger, so it will hold more, carry more what? Water. Okay, it will carry more water, or it will contain more water, or it will store more water, because we are not talking about oxygen transport. We are talking about dehydration and storing water. So this shape may help it to store more water. Something else. Give me another way this shape or this red blood cell will be more adapted in dehydration. So we will say the biconcave shape of the human red blood cells, it will store less water. because any cell, of course, contains water. So it will store or hold less water. Something else.
Doctor, my answer is I'm not completely sure, but for both of them, they for the same يعني, area, there is more red blood cell in the camel.
Yes. By the way, Mariam, you have to understand that in suggest questions, you will never be sure of the answer because it is a suggest question. Everyone is going to suggest something different, and it is a critical thinking question. Yes, I also see it. It's very obvious. The two things that you see in those pictures, the shape difference and the number, it's obvious also. It's obvious that here in the camels, they have more red blood cells. And in the same space inside the human blood, I have less red blood cells. You're right. This is what we can see. So we can see differences in shape and we can see differences in number. This is one of the most obvious differences. Okay, so how will this help in dehydration? So the camel red blood cell, they are more in number,
Therefore, more
and
red
in
blood
humans,
cells carry water.
very good. So they are less in number. So more red blood cells means they will carry or store more water. And the human less blood cell, they will carry less water. Now, this is out of 3 marks. So you have to think of a third difference. What else can you see between both? I can see also something, then I assume that it will help, but I want your ideas, not mine. You can see that this is perfectly oval. It's a little bit larger, and they are more in number, so they will hold more water and help in dehydration. What about the cell membrane here and the cell membrane here? What will you say? You have to say a third point. Farouk, Adham, Kareem. You did not say anything, Kareem, today. What will you say different between the camel and the human red blood cell?
واحنا ما نقدرش نقول ان في نيوكليوس عشان احنا مش شايفين اللي جواها.
Exactly. No, you can say that. Why? Because obviously this is biconcave. There is a depression in the center, which means that there is nucleus or no nucleus?
No nucleus.
No nucleus. This is definitely, you're definitely sure that there is no nucleus here. Here, you do not see a depression in the center. So there's a very high chance that it does have a nucleus. Okay? And if it does have a nucleus, how can this help in dehydration? But how can this help in dehydration?
It has a nucleus, so can it help in dehydration?
Does it help in dehydration?
No.
It doesn't really. Actually, when you go and search it, red blood cells in camels, they do have a nucleus, and it's there for other reasons, not for holding water. But you can say something else. Forget the nucleus now. Yes, red blood cells, they have no nucleus, and the red blood cells in camels, they have a nucleus. But we don't really know how this will help in dehydration. The cell membrane of the red blood cell in camels, it is very clear. The cell membrane might be thicker or might be stronger. The red blood cells inside camels, they have a very high elastic power. They can stretch and they can hold 20 times more water than the red blood cells of the humans. The red blood cells of the humans, their cell membranes are very thin and fragile and they easily burst. Okay, this is something that it's not very, very obvious, but this is something that you have to think of. I don't know if you wrote only those 2 points, will they give you the 3 out of 3 or not? But as long as you have a question out of 3 marks, then most probably you have to write 3 points. We obviously notice the difference in shape. So this is oval and this is biconcave. This is bigger, so this will carry more water. Mariam noticed that they are more in number. Yes, this is a very obvious difference. They are more in number. so they will carry more water. Now, a third difference. These are biconcave, there is no nucleus. And these have a nucleus. But how do they help in dehydration? I cannot think of something. What you can notice is that the cell membrane here is very clear and very obvious, and it might be thicker. And the red blood cells of the camels, they can stretch much more than the red blood cells of the human, and they can hold more water without stretching. That's it. Are you with me?
Yes.
Okay. Now, I did notice when I looked at this exam, the cell membrane part, and then I searched it out and I realized that it is true. The red blood cells of camels, they do have a nucleus, yes, but what really helps in the dehydration is that they can store more water because they're oval, and they have a cell membrane that is stretchable. It can stretch. Now, this is something that I do not expect you to know. Maybe in the picture, the cell membrane is just obvious and it's prominent. And here the cell membrane is not very prominent. And maybe you will mention something like this because you already know that the red blood cell, it will burst in concentrated plasma. I hope it crosses your mind. But so we'll write it down that the cell membrane is thicker. So it will stretch when it is filled with water. The red cell of the camel here is stretchable, or it is more elastic. The red blood cells in humans, they burst when they are surrounded with concentrated solution. The first two, you would definitely get them. And maybe the first two will be enough. When you mention the first two and elaborate, you will get a 3 out of 3. The third one, I doubt you will get it. Maybe it can cross your mind, maybe it will not. Okay, now here there is an oxygen dissociation curve with the partial pressure of oxygen on the x-axis and the percentage saturation of hemoglobin on the y-axis as usual. And you have a key. You have the solid line for the adult hemoglobin for the llamas, and you have the dashed line for adult human hemoglobin. With reference to the figure, explain how the differences between the oxygen dissociation curves for humans and llamas show that llamas are better adapted for living at high altitudes than humans. 3 marks. Who will answer this question? This is also a very easy question. Why are llamas better adapted to live at high altitudes? This is very easy.
Because as the partial pressure of oxygen increases for both the human and the llama has a higher percentage saturation of oxygen, of hemoglobin with oxygen than the
Yes.
adult human. Therefore, the red blood cells have a higher affinity to oxygen and can carry more oxygen around their blood.
Very good. Perfect. They have a higher affinity to oxygen. The red blood cells. So llamas, the red blood cells, they have a higher affinity to oxygen. This means that they carry more oxygen at the same partial pressure than humans, just like Mariam said. And what did you say? What was the last sentence you said? You said that they can carry more oxygen. And why is this an advantage? Because what is the problem with high altitudes?
There is
Oxygen
less
more.
Oxygen is more or less?
less.
Yes. To compensate for the less oxygen at high altitude. So this is a very easy question. Very, very easy question. Up there at the mountain, I have less oxygen. So llamas, they can live up there. They will have no problem, but not us, because llamas, they already have a hemoglobin with higher affinity. So the same amount of hemoglobin will carry more oxygen. Very good. Humans, if they go and live at the top of the mountain, they cannot survive at the top of the mountain? Or they can?
They
What if
can.
they change the question? They can. They only need some time for their bodies to adapt to the change in the environmental conditions. Yes? What will their body do in order to adapt to the high altitudes?
Produce more red blood cells.
Correct. The affinity will not change. But what will change is that we will produce more red blood cells so that we have more oxygen, so that we compensate for the lack of oxygen at the top of the mountain. So people that live at the top of the mountain, they don't have more affinity. They just have more red blood cells. So they can carry more oxygen at the same volume of blood. So they will compensate for the lack of oxygen. Sketch a curve on the figure to show the effect of increased carbon dioxide concentration on the percentage saturation of adult human hemoglobin with oxygen. Okay, we need to stretch a curve to show the effect of increased carbon dioxide concentration on the percentage saturation. How will you sketch this curve? Where is Kareem? Kareem, you're here with us, yes? Now you are in a situation where you have more carbon dioxide. How will this affect the percentage saturation of hemoglobin with oxygen? The curve will go up or down?
Up.
What do you think? I want everyone else to tell me, what do you think with what Kareem said? Will the curve go up or down? Will the hemoglobin have more affinity or less affinity?
Less affinity.
Of course, less affinity. Do you all agree? Of course,
Yes.
less affinity. You have more CO2. So you will have more reaction, CO2 plus water, to give me carbonic acid. And this will dissociate into hydrogen ions and hydrogen carbonate. And the hemoglobin will bind with the hydrogen ions to give you hemoglobinic acid to prevent the decrease in pH. So the hemoglobin will have less affinity to oxygen. So the curve should be anywhere below, something like this. Okay? Kareem, are you with me?
Yes, miss.
Explain the importance of the Bohr shift in metabolically active organs such as the liver. Now, this is an important question. I just gave you the answer, actually. Why is the Bohr shift important in active organs? First, what is the Bohr shift? I don't know why you don't give me an answer. What is the Bohr shift, and what does it, and what is its importance, and how does it shift when you are exercising or in active organs, and why does it shift? We did not take this. We did not mention this 100 times. Adham,
Yes, miss.
what is the Bohr shift?
The Bohr shift,
Yes.
when the graph of the partial pressure of oxygen shifts either to the right or to the left.
Very good. Why didn't you give me an answer? Very good. Bohr shift, from the name. It's the shifting of the oxygen dissociation curve. So it is the shifting or the movement of the curve. Just like Adham said, shifting of the oxygen dissociation curve, either up or down, just like Adham said. Now, when you have active organs means what are they doing?
Respiring.
Very good. We are respiring. Let me write it down. There is respiration. So what are they producing?
Carbon dioxide.
Very good. An increase in the concentration of carbon dioxide in the blood. How will this make the curve shift, and why?
It will shift to the left.
Right. Yes. Wait. Shift to the right. Up or down?
Up or down.
If the curve looks like this, it will move up or it will move down. When I have more CO2, down. It will move down. It's just down is more clear. Now it will shift to it down. What does this mean, and why is this important? Well, how will this affect the affinity of hemoglobin
Means
to
that
oxygen?
the partial pressure, the affinity of hemoglobin to oxygen decreased, and that
Correct.
the percentage of oxygen that's bonded to hemoglobin is also decreased.
Correct. Perfect. So the hemoglobin affinity to oxygen will decrease. The hemoglobin will bind with what instead?
Carbon dioxide.
Very good. Now the hemoglobin, we said it is always, no, not carbon dioxide. The hemoglobin binds to what?
Carbon monoxide.
No, ya habibi.
Hydrogen.
Yes. The hemoglobin is always bound to something. The hemoglobin is always bound to the oxygen. For example, it will give me oxyhemoglobin. But now I have increase in the CO2. The CO2, write the equation down in this question. The CO2 reacts with water in the presence of carbonic acid to give me carbonic acid H2CO3 that will dissociate into hydrogen ions and hydrogen carbonate ions. Now, the problem is with the hydrogen ions. What is their problem? Why do we want to get rid of them? Why does the hemoglobin bind with them?
Because it has higher affinity to hydrogen than oxygen.
Yes, they do have a higher affinity, but because if we leave them in the blood, what do they do? How do they affect the pH of the blood?
It will increase because it forms hemoglobinic acid. So the pH is lowered.
Yes, it will lower the pH. So to prevent a decrease in blood pH, the hemoglobin will dissociate from the oxygen. I will detach. This is called dissociation or unloading. This oxygen will go to the respiring tissues, and the hemoglobin now it will be free to bind with the hydrogen ions to give me hemoglobinic acid. And the hemoglobin here will act as a buffer. And this is very important to prevent a decrease in blood pH. Okay, so you will write the whole story. Again, this is the direct question, by the way. Maybe just you will mention the Bohr shift, and Adham easily said it is shifting of the oxygen dissociation curve. So this is an easy question. You studied well, you can answer it. You did not study well, you will not do a good job in this question. Okay, any questions? Up to now, the exam is a little bit harder from the previous exams that we answered before, a little bit. Now, this is a picture of what? Let us see. Figure 3.1 is a picture showing part of a transverse section through the root. This is the root of an iris. Irises are herbaceous monocotyledons. These plants have the same transport tissues as the herbaceous dicots, but the transport tissues are distributed differently. In monocots, the central tissue in the root is parenchyma, packing tissue. Okay, so this is here, we are in the center. I want you to put this in mind. We are in the center. The vascular tissue, the xylem and the phloem is in the center. I said it 3 times. Name the tissues in which the cells labeled R, S, and T are found. What is R? It's the center, the center, the center. So what is R? Who will give me an answer? What's this? I lost connection. Hello? Farouk, what is R?
Miss, I can't see the full cell, but mostly you have to picture it's perfect. Like
Is it
last
now
time.
better? Okay, what is R?
I can't see.
Okay, I'll share. I'll stop sharing and then I'll share again. 1 minute. What is R? Can you see now the screen? Everyone, Farouk, can you see the screen?
I can see.
Yes.
Okay, what is R?
Well, it's not very clear. Printing is not very clear, but I think vascular bundle.
No. The vascular bundle is in the center. This is the vascular bundle.
Okay.
And they said that this is the central tissue. So and they said that this is the root. So we have a root here. We have a root, and this is the central tissue. So this is the root. Outside here, we have the epidermis and the root hair cell. And then we have here another layer. And within this layer, we have the vascular bundle. So this inner layer, what is it? What is R?
The inner layer.
I'll give you 2 options.
I can't say
Epidermis or endodermis?
يعني I would have said endodermis, but it says cells.
Yes, you are right. It is the endodermis. Yes,
Okay,
these
cells.
cells make up the tissue in which cell R is found. It's an endodermis.
Okay, I said يعني أصل هو كاتب cells are I thought it was a cell يعني بس it
Any
is an
tissue is made up of similar cells. So this
okay.
R is the endodermis, not the epidermis. Now, this is a tricky question. Okay? This is a tricky question because a lot of you will say epidermis, and because the image here is zoomed in, the epidermis is not in the image. We have an external epidermis, and then we have an endodermis that blocks the transport of water, and then the water will change pathways, if you remember, from apoplastic to symplastic, and then it will reach the central xylem. What is S? This big tissue, what is it? Xylem or phloem? What is S?
Xylem.
Xylem. And it is surrounded by the smaller T. What is T?
Phloem.
Phloem. Okay, again, this is a tricky picture because this is a monocot, not a dicot. We are used to dicots, which has the external epidermis, and then we have the internal endodermis, and the dicot, the vascular bundle looks like a flower. The central tissue is the xylem, and then it is surrounded by the phloem. But they told you in this question that it is a monocot. If you read the question carefully, they tell you it is a monocot, and the central tissue in the root is parenchyma, and the tissues are distributed differently. Okay, you do have pictures of monocots in your notes, but this is something that you will not also notice, except if they had told you. They already told you, so you don't have an excuse. So the tissue T is a monocot, and the S is an xylem, and the layer that surrounds the central vascular tissue is always the endodermis. Outline the role of the tissue in which cell R is found. So what is the function of the endodermis? 2 marks. What is the function of the endodermis? In lesson plans. Don't stay silent. Give me an answer. Hello? What is the function of the endodermis? It is in your notes, and we did say it in class. I checked myself. What is the function of the endodermis? Who remembers? You will all stay silent? What happens at the endodermis? By the way, I said it while I was explaining. Hello?
Simplest pathway.
Yes. The water changes pathway from what to what?
Apoplastic to symplastic pathway.
Okay, very good. And there is a function of, this is one, it controls the pathway of the water. Another function of the endodermis, written right here. What is it?
Physical barrier preventing water loss from the tissues it surrounds, as well as providing protection against pathogens.
Yes. Okay. This sentence, the answer is this sentence. Please study every single word. So going back to this exam, what is the role of the tissue? Number one, if you do not know what is R and you got it wrong, you will get this question wrong. That is why this question is tricky. So I will highlight this question. This is the endodermis, and it has several functions. We'll write the functions down now. First, it will control the pathway of water. One, control water pathway. Just like Farouk said, it will change the water pathway from apoplastic to symplastic. Two, it provides protection It prevents the entrance of pathogens from reaching the central vascular bundle, the phloem. B, it also prevents the loss of water. You can write that. Prevent water loss. So I recommend that this is an important question and please pay attention to it. Okay? Fine. State an example of an organic compound that is translocated in the root of any plant. What is translocated in any translocation here happens where? Phloem, xylem. What is translocated? Give me an answer. Can you please be quick? I'm not going to answer all the questions for you. I'm not going to be the one sitting for the exam. It's going to be you. So what will you say in this question? What is being translocated in the phloem?
Sucrose.
Okay, give me an answer. Don't stay silent. Sucrose, amino acids. Here, an electron micrograph shows a section through some root hair cells in an onion. Draw a label line and label it with the letter P to identify one plasmodesmata. The picture is not very clear, but the plasmodesmata is going to be, of course, 1 minute. Okay. The plasmodesmata, it's not very clear, but it's going to be here. Somewhere here. Anywhere on the cell wall of the plant. Any vertical line here is going to be the plasmodesmata. So I'm going to put a letter P here. Continue this table. The amylose. What is the monomer in the amylose?
Alpha glucose.
Alpha glucose, yes. Cellulose, the monomer is beta glucose. What is the glycosidic bond here? In the cellulose.
1 to 4 glycosidic bond.
Okay. Not because I told you to answer, Mariam will be the only one to answer. Everyone participates. Glycogen. What is the function of the glycogen, Farouk? Farouk.
A store of energy.
Yes, it's an energy storage. Where? In plants and animals.
Yeah, your voice is lagging. Just give me a second.
It is energy storage in plants or mammals? Or
Mammals.
animals?
Animals.
Animals, yes. And fungi. So I'll write it down. Animals and fungi. Okay. I will not read the question. This is the DNA template strand, and this is the primary transcript. Here we're talking about RNA, of course. Okay, tell me, Adam. Is Adam with me? I don't know, where did he go? Kareem. Kareem? C.
Yes, miss.
The C. What is complementary to it on the messenger RNA in the primary transcript?
I can see the screen.
Is the screen visible now to everyone? Can you see it now?
I only see Dr. Manashul Kami is presenting.
Okay, stop presenting.
Yes, I can see it now.
Okay. Now the bases that are complementary, adenine binds with thymine or uracil and RNA, and cytosine binds with guanine. Now C will bind with what on an RNA? What's the complementary base?
G.
C binds with what?
G,
Yes, G.
guanine.
Yes, A binds with
T,
on an RNA, thymine or uracil.
RNA, uracil.
Yes. And the C, we said G.
G,
Now T binds with what?
A,
Yes. A, we said uracil. C, G,
G,
T,
A,
A,
G,
C,
G,
G, G,
U,
T,
U.
A, U, U. Yes, correct. You're right. Because this is an RNA. And CG. Good. Okay. Now here we have the amino acids and the different DNA triplets that code for the amino acids. Complete table 4.3 to identify the 4 amino acids labeled AA1, AA2, and 3 and 4. So AA1 is which amino acid? Let's go to the first table. AA1 is CAC. CAC is going to be valine. So we will write VAL here. AA2, AA2 is TAC. Where is TAC? It's going to be this one. MET, MET. AA3, are you all with me?
Yes.
AA3 is TCC. I'll go to TCC, I'll find it here. ARG. AA4
Leo.
is AAC. AAC, where is it? This one. So it's going to be LEU. So this is a very easy question up to now. Okay. Using the information in table 4.2, state and explain the effect on the final protein structure of a substitution of the nucleotide at position 3 in table 4.1. Now, this question needs a little bit of concentration. You will go to table 4.1 first, and you will go to position 3. So where is table 4.1? This is table 4.1. You will go to position 3, and you will tell yourself that this is going to be substituted, this one. It's going to be substituted. Now look at the DNA template. It is CAC. CAC can change into CAT, CAG, CAA. So if it changes from CAC to CAT or CAG or CAA, what will happen to the amino acids?
Nothing.
Nothing, exactly. Which type of mutation is that?
Silent mutation.
Excellent. So when we use the information, you will say that no changes will happen. This is a silent mutation. In all cases, the base will be substituted, yes, but the amino acid valine or the VAL amino acid will be produced in all cases. Now, why? Why, even though there was a mutation, but no change happened?
Because the genes are degenerate, therefore the
Yes,
same amino acids have multiple base triplets.
very good. This is because the base triplet in this case is degenerate. Perfect. Now, a second type of mutation is caused by the deletion of a DNA nucleotide. Using the information in table 4.2, state and explain the effect on the final protein structure of a deletion at position 3. So here is a different story. Faru, are you with us? Who do I have?
Yeah.
Okay, tell me, Faru, if nucleotide number 3 has been deleted, it's not there. How will this affect the protein?
The first amino acid would be GUA.
It will be, don't look at the primary transcript, look at the
No,
DNA template because this is
no,
what we have here. So it will be CAT instead of CAC.
Yeah, it will be CAT. So,
Huh?
so it will change from CAC,
Which is,
from YAL, YAL, to it won't change. Nothing will happen.
it will
The
not
first,
change. But what will happen to the remaining base sequences?
they will shift.
Very good. Very good. So what will you say in this question? In this question, you will say that one of the base sequences will change or they will be deleted. Like they said, this is a deletion mutation. The first amino acid, like Farouk said, it will not change. But what will happen to the remaining amino acids and the remaining base sequences? Farouk also said that they will shift. They will shift to the left, okay? And what are the names that we give to these type of mutations? There are keywords here, Farouk, that you will mention. You said that they will shift. All the bases will shift. You are correct. What is this type of mutation called? Or what effect does it have?
It affects the whole polypeptide chain.
Yes, you are correct. It affects the whole polypeptide chain. All the amino acids from the point of mutation onwards will change. So what is the keyword that I'm looking for? Let me remind you, it has a
A knock-on effect.
very good. It has a knock-on effect. And this type of mutation is known as the frame shift mutation. Yes, didn't you say that the base is going to change and all the remaining bases will shift?
Yeah.
Yes, and all these sequences of the bases will change from the point of mutation onwards. And this is called a knock-on or a frame shift mutation because all base sequences will change from the point of mutation onwards. And just like you said, all the amino acids will change from this point onwards, and the final protein will change or it will remain the same. Yes, Farouk, the final protein will be the same or we will create a different protein.
No, it will be different protein.
Yes. Please don't forget to mention everything. Draw a circle around each event where replication of nuclear DNA occurs. Nuclear DNA, where does it take place? In which one of those?
I missed the screen
Now this,
sharing.
again?
Yeah.
Adam. DNA replication happens in which step here?
S phase.
Yes, S phase. Can you choose something else? You can choose another
Interphase.
interface. Yes, easy. This question, I will also highlight it. Okay, so this exam doesn't finish? It doesn't finish. It's a long exam. Outline how DNA is replicated inside the nucleus. Now, this is the direct question. I'm going to leave this question, you answer it. It is the very direct. Just copy paste it from the notes. State the name of the part of ATP molecule labeled A. What is A?
Ribose.
It is the ribose, not the deoxyribose. It's the ribose. Okay. Look at carbon number 2. Here you have a hydroxyl group. Hydroxyl group in carbon number 2 will make it a ribose. So wait, I'll go to this question. This is a direct question. I'll write here direct question. Just write the steps of the semi-conservative replication. All the steps. Don't leave anything out. Okay. Finally, the last question. Name the type of electron microscope used to produce this image. What are the two types of electron microscope? Mariam, this is which one of them?
Electron microscope and the scanning electron microscope.
Yes, this is which
Yes,
one?
the
Can you
scanning.
see inside it? This is scanning, correct. You said scanning, yes?
Yes.
Yes, this is scanning because I cannot see inside the bacteria. I can only see the outer image. It's a 2D image. It's a 3D image. Name the species of the prokaryote. I just told you. What is the species, the prokaryote that produces cholera?
Bacteria.
It's a bacteria. Okay. Prokaryotic cells are unicellular and generally between 1 micrometer and 5 micrometer. Prokaryotes do not have organelles surrounded by double membranes. They do have a cell surface membrane, 70 Vedberg ribosomes, and a cellulose cell wall. The DNA of a prokaryotic cell is circular and it is found free in the cytoplasm rather than enclosed in a nuclear envelope. Tell me, Farouk, identify and correct the factual error in the passage. Give me one clear error in this passage.
It doesn't have a cellulose cell wall.
Very good. The cell wall is made up of what?
Murin.
Yes, murine, or it has another name, peptido.
Glycan.
Very good. So this is an obvious mistake. Okay. Identify, Adham, identify structure X in the virus. What is X?
Capsid.
Yes, correct. It's the protein coat or the capsid. Made up of subunits called capsomeres. Okay, the protein coat. Correct. Now explain how the ability of the immune system to resist the damaging effects of a pathogen is affected by destruction of the T helper cells. Again, this is a direct question. The T helper cell is destroyed. How will this affect your immune system? For the 10th time, for every exam, this question has to pop up. Immunity question and the action of the T helper cells. We said it so many times. I'm not going to say anything. When the T helper cells are destroyed, what will happen?
No primary immune response because there is no
Excellent.
clonal expansion or clonal selection and no dividing by mitosis. So no primary immune response, and therefore the human body is more prone to opportunistic diseases.
Correct. Everything you said is correct, but you missed one small step. The first step. T helper cells, what do they release?
Cytokines.
Yes. So you will only remember the normal function of the T helper, and all this function is going to be affected. And everything you said is correct. No cytokine, no colonal selection, no colonal expansion of lymphocytes. Okay. No production of antibodies. If, for example, we're talking about the B lymphocyte, no production of antibodies. If we're talking about the T killer cells, no direct killing of infected cells because they will not work. No activation of more macrophages. No immune system, and you will be liable to opportunistic infections. Everything you said is correct. Perfect. Okay, so please, this question, please study the steps of the immune response. It comes in every single exam. Suggest how mutation of the gene receptor protein provided protection. Now we have to read the question. Studies have shown that some individuals did not become infected with HIV, even though they were repeatedly exposed to the virus. They have immunity to HIV, they're lucky. Later discoveries included that these individuals had a mutation in the gene for the co-receptor protein. Suggest how a mutation for the gene for the CCR5 co-receptor protein provided protection against HIV. So when you have a mutation in a gene that is a receptor for HIV, how will this protect you from HIV? Very easy question also. Farouk, how can you answer this question?
Give
The receptor that receives or allows the entrance of the HIV into the host cell has been mutated.
Okay, so the HIV can't enter anymore, so it can cause an infection.
Yes, but we will not write that. We will say that the receptor is no longer
Complementary.
very good. So the co-receptor will become mutated. The shape of the receptor will change, and it is no longer complementary. And the HIV cannot infect the host cell. Okay? It's an easy question, but they like to ask the question in a twisted way to confuse you, but it's an easy question. Outline how anti-CCR5 monoclonal antibodies can be synthesized in the lab using the hybridoma method. Again, this is the direct question. Who will remind me, what is the hybridoma method? This is the
You
last question.
include a non-foreign antigen in a foreign antigen into a mouse,
Very good.
and
And then what?
stimulates the immune response of the mouse.
Yes.
And then it produces plasma cells in the spleen.
Very good.
You
Huh?
extract the plasma cells from the spleen of the mouse and
Yes.
fuse them with cancer cells, myeloma cells, using
Correct.
the oxygen, like electricity, and then
Very good.
you humanize it.
Excellent. You will form a hybridoma cell. And then what did you say? I'm going to humanize.
Yes.
Humanize. And then finish?
Then you test and screen it.
Yes, very good. I will test that it is producing the desired antibody. I will screen it. And then what? I screened it and I'm sure that now it is producing the antibody that I want. And then I'm going to put it in a fermenter. I'm going to clone it. Yes.
Yes. Okay.
I'm going to clone it so that I produce more of this hybridoma cell and I will extract the final antibody and I'm going to use it in passive immunity, in diagnosis, in a lot of things. Okay, so again, this is also a direct question. Who will remind me, how do we fuse the myeloma
Use it.
cell with the plasma cell? Fusion,
Like electricity.
polyethylene glycol, or a small electrocurrent called, just a small electrocurrent is enough. We finished the exam. Now this exam, if you noticed, we solved many exams and the past exams, the 2023 exams, they were easier. Didn't you notice that? This exam is a little bit tricky. It's in like the level of hardness is increasing and this is not good news. Maybe it's good. This means that the curve will decrease, but it's going to make you panic on the exam. This question was easy. This question also was very easy. Cell membranes. This question, I did not. It was a direct question also. Cell signaling is very. This question was not nice. It's a suggest question. Suggest questions are always not nice. Put this in mind. So suggest questions, always know that when you get a suggest question, it's a critical thinking question. You will not find it easy. It's tricky. And you just have to do your best on the suggest question. So this question, I highlighted it so that you look at it again. And this question is easy, but if you did not study the lesson, you will not get it at all. The oxygen dissociation curve. Okay? And the Bohr shift. So it's a very direct question, but if you did not study the lesson or you ignored it, you will lose the whole question. This question, I thought it was tricky because I also said that they will not be able to remember the endodermis, but even though they asked you this question, when they ask you for the function of R, hopefully you will remember that it is an endodermis. But this question here, please go to the lesson plans and look at all the pictures that you have in the lesson, because you will find pictures of the root and the stem. Please look closely at the pictures. The pictures are not there to waste space or waste ink. The pictures are very important. Look at the pictures in the two lessons, respiratory system and the plants. The pictures in your notes, and go to Google and look at more pictures. The pictures in your notes are never enough. The pictures in any book are never enough. Go to Google and write in the search section, I want to see a cross section of a monocot root. I want to see a cross section of a dicot root. Get familiar, be familiar to the pictures, because as you can see, they always get you pictures on an exam, whether paper 3 or paper 2, and they ask you to label. You always have a problem in differentiating the smooth muscle and the cartilage. You have a problem with differentiating the different tissues in a plant. So please go, while you are sitting free, go to Google and search microscopic pictures of plant tissue and respiratory tissue. Okay? And get used to the pictures. And of course, they will give you the pictures with labels. This will really help. This was easy. This was easy. This was very easy. Okay, I highlighted those two questions because Here we're talking about base mutations. If you did not study it, you will not get it. This is a direct question, DNA replication. This was a tricky question. Okay, you have to really know the difference between the ribose and the deoxyribose sugar, and the rest, it was really easy. Monoclonal antibodies, and it was a direct question. Okay, the March 24 exam, in my opinion, you will not do very well on this exam because it's a little bit tricky. More than one tricky question. This is showing us that the exams are getting harder. We need to study more. Any questions before we go?
No, thank
Hello?
you.
Thanks.
Doctor, thank you.
Okay, we will meet on Monday in school. And please study harder. How do I know that you studied well? When you give me quick answers, when you don't spend any time thinking, when you sit and think, this means that you need to study harder. You forget. I don't want you to forget things. And again, like you're doing, if you have any questions, send me on WhatsApp, okay?
Okay.
Okay, I'll send you the exam now. Bye-bye.